Day of Faculty Development Program. Myself, Dr. K. Swati Priya, co-convener of the Faculty Development Program on the theme Challenges and Innovations in Advanced Pharmacognosy, organized by Srinivasarao College of Pharmacy. Now, I request Mrs. Kankadurgavalli ma'am, HOD of Pharmaceutical Chemistry Department, to introduce our today's resource person.
Good morning, one and all. Myself, Kanasagutavani, HOD of Department of Pharmaceutical Chemistry and IQAC Coordinator. Welcome you all for the second day of FDP program conducted by Srinivasarao College of Pharmacy. And it's my privilege to introduce our today's resource person, Dr. R. Lakshmi Marsetti. She was born in Andhra Pradesh and she is currently working as Assistant Professor in Department of Pharmacognosy and Phytochemistry, Pharmaceutical Sciences and Research University, New Delhi. She completed her MPharmacy in 2011 and doctorate in 2021 in the Department of Pharmacognosy and Phytochemistry from Andhra University, Visakhapatnam. She is having 12 years of teaching, research, and administrative experience. She has guided 26 B.Pharm and 7 M.Pharm students. She is awarded Best Outgoing Student for the academic year 2005 to 2009. She secured state first rank in ASET 2017 and Best Poster Award and prizes from various organizations. She has published 29 research papers in various national and international journals. She has 7 patents of India and 2 international patents. She has 2 ongoing projects under DST, SERB, SURE, and CCRUM, Ministry of OIS, GOI. She is nominated as Young Professional OIS 01 and OIS 01. Ayurveda Sectional Committee and Unani Sectional Committee, Bureau of Indian Standard, New Delhi, India, 2023. She's having membership in various national and international professional societies, and her area of research interest are isolation and characterization of phytochemicals, pharmacognostics and phytochemical analysis, biological activities, natural product pharmacology, ethnopharmacology, traditional medicine, herbal medicine, and herbal drug development and standardization. Welcome, ma'am. I request you to proceed to the session. Thank you.
Thank you so much, Valli ma'am. Thanks for the nice introduction. Myself, Dr. Arya Lakshmi. Can you please share my slides? Myself, Dr. Arya Lakshmi, working as an assistant professor in the Department of Pharmacognosy and Phytochemistry, Delhi Pharmaceutical Sciences and Research University. Today, we will discuss about the modern herbal medicine, the necessity of standardization in today's healthcare system. Next. So we all know that herbs are the crude plant materials such as the leaves, flowers, fruits, seeds, and which are obtained from the various parts of the plants, which may be used as such that part, or we have to fragment it or the powdered. And we have to know about the herbal drugs. Herbal drugs are the finished label products that contain active ingredients such as aerial or underground part of the plants, and we have to use the as such the material, and we have to combination of the various plant materials, whether in the crude state or in the processed state. Next. So we all know that Vedas are our secret knowledge of. We have 4 Vedas: Rig Veda, Samaveda, Ajurveda, and Atharvan Veda. So we all know that God revealed the various secrets. Among all, Atharvan Veda contain the maximum amount of the medicinal secrets which can be used for the various diseases. Uh, Majority 88 percentage of the plants of the world population reveals the all the traditional medicines are used by the throughout the world. 88 percentage of the all population reveals on the plant based traditional medicines. And why we have to go for the herbs? Because nowadays, after post COVID era, everybody is most concentrating on the herbal medicine and herbal drugs because they are more safe and no side effects, low technical input, and easily accessibility, and increase the complexity along compared with the modern medicine, and they are more effective, affordability, and we have the flexibility. Then now we are in 21st century, is the century of the biology powered and propelled by the scientific knowledge. So we have the technology expertise. So we all know that we have three technologies: biotechnology, herbal technology, information technology. So our main target is, as a pharmacognosist, we are more concentrating on the herbal technology, the most powerful element that are crucial for properties and welfare of the mankind. So we have to know about the various co-branches in the herbal drug technology, like herbal drugs, nutraceuticals, biopharmaceuticals, and phytopharmaceuticals, cosmetics, etc. So before, we have to discuss about various standardization protocols and why we have to go for the standardization of the herbal drugs. We have to know about the revolution of the herbal drug technology. Then we have four revolution, and in the first revolution, and we all know that we know during our in the Ramayana, pre-Vedic period, we for the when Lakshmanji got injured, then Hanuman brings on Sanjeevani Mulika, then he used it to treat him. So we know that before Vedic culture also, we are using the herbs as a medicine. So during the pre-Vedic period, like Rig Veda, Atharvana Veda, Samaveda, and these Rig Veda having a little amount of the knowledge regarding the medicinal plants, but among all, Atharvana Veda giving the clear picture regarding the what are the drugs, what are the plants we are used for the treatment of the various diseases of the mankind and also the animals. And post-Vedic period, we all know that Sushrita Samhita and Charaka Samhita, these are the treaties are given some valuable uh, plant properties of the plants and make their various therapeutic properties in that post Vedic culture. And coming to the second revolution, we started plant drugs from the wild sources, and according to the demands of the demand of the world population, they started for the cultivation of the medicinal plants as well. In case in the first revolution, identification, no, backslide, identification of the plants, we have to go for the various documentation like Vedas, and also various organoleptic characteristics. In the second revolution, we are going for the ethnopharmacological and the traditional knowledge. We have to procure the knowledge from the various traditional healers and from the various siddhas and yogis. We have to identify the plant. And in the first revolution, we have to use various gutikas, churnas, and also, and in second revolution, we started preparation of the gutika, avalekya, aristas, and various formulations are coming to the picture in the second revolution. And in first and second revolution, there is no regulatory guidelines for the plant related preparations and the medicines. In second revolution, drug and cosmetic act came into the picture for the standardization protocols for the herbal drugs. Next. And in the third revolution, the sources are, we are preparing various herbal extracts and herbal depending upon the property of the chemical constituents which can be used as a potent drug. We have to potent egg, we have to use various solvents accordingly. We have to start the extraction process. From the identification, we have various microscopic characterizations, electronic microscopes, and various characterization of plant powders, and also various parts of the powders, like we have to identify while comparing to the organoleptic property, we have to identify more and more properties of that particular powder and crude drug material. While coming to the preparation, there is various sophistication instrumentation technologies all there. Various extraction procedures came into the picture in the third revolution. Here soxylate apparatus, and also microwave extraction, and Also various high throughput screening lead molecules. We have various biotransformation properties also, according to we have to change the molecule and we have to incorporate into the another chemical. We have to go with the another phytoconstituents which is having the which is having the reduce the adverse drug reactions. And here also in regulations, US FDA, Australia, European Union, China, India, various regulation guidelines came into the picture in the third revolution of the herbal technology. Next. And this is now the fourth revolution. Here plant tissue culture is came into the picture for various endangered and threatened species. We have to cultivate it, we have to develop in the plant tissue culture, and also we have to develop secondary metabolites, those who are having the potent activity in that plant, that we have to increase by using various precursors and elictors. In that plant tissue culture, we have to go for the plant biotransformation also. And for identification, various technology, sophisticated microscopes also available, conifocal microscope and SEM, various identification methods also came. And for the preparation also, we have various advanced technologies are there, plant genomics and reverse pharmacology. And here in regulatory standardization of herbal raw material and finished products by the Asian regulations. In this our in the fourth revolution, we have to make a individual single regulatory guidelines for all the herbal medicine, not only for the depending upon the various countries. Now the Asian regulation, it can be throughout the Asian countries, they have given us some protocols for the standardization of the herbal raw materials. Next, after post COVID revolution, we have uh, we have to cleanly identify the genomes, and we have to coming to the green biotechnological revolutions, and also oil genomics. And we have to identify the problem in the genetical level, molecular level. We have to treat the diseases. And coming to the regulations, recently in 2022, and our India is declared as a global center for the traditional medicine. We have the some collaboration with the WHO. Our prime minister started this in Gujarat. So the aim of this WHO global traditional knowledge, we have to came into the picture along all the traditional systems of medicine. We have to develop, and also we have to accepted by the Indian systems of medicine. Next, herbal drug market in India. The value of the botanical related trade is about various 10 billions, and China, it is the cost of 48 billions. It may be increased or in the year of 2050. Next. And coming to the Indian systems of medicine, our Indian systems of medicine, we all know that Ayurveda, Siddha, Unani, it is from the 1000 to 500 years BC. Because why we have to go for the Indian systems of medicine? This is acceptable, affordable, and more effective. And coming to the Indian scenario, why the traditional systems of medicine is compared to the all the traditional systems of compared to our Indian systems of medicine is the second place, while compared to the China systems of medicine. 2.4% of the old they're using, and Indian biodiversity 88.1% of the world, and total 45,000 plants are used as a drugs and medicines in the Ayurveda. And we all already identify the 45,000. Among 45,800 species are used in Ayurvedic medicine. And we have Indian Ayurvedic formulations, 3,000. We are using Basmas. Indian registration till now, and 6 lakh Ayurvedic practitioners are there for treating various diseases according to the Ayurvedic systems of medicine. All and overall 42 crore of Indian systems of medicine in the industry, Ayurveda contributing 35 crores, and also with about 7,000 manufacturings of Ayurvedic products. Not only Ayurveda, we are also having the Unani medicine and also Siddha systems of medicine also available in our Indian systems of medicine. This is the Indian systems of medicine market in India: Ayurveda and homeopathy, Siddha and Unani. and among all Ayurveda leading 84 percentage. And globally, the Indian systems of medicine globally, among all Asia having 18 percentage world market, it can be occupied 18 percentage of Indian systems of medicine in the world population, in the world market. And also a growth potential, Indian systems of medicine is generally called as complementary and alternative medicine outside India, while is referred to as under the name of traditional medicines by the WHO. So the economic importance and use of the herbal medicines is increasing worldwide, in that 15 percentage is compared only the 3 percentage of the modern medicines. So herbal drugs are best studies for herbal, not only for nowadays. Previously in Ayurveda system they are giving one drug for the whole system of the, now they are having the individual, they are using for the various infectious diseases, geriatric patients, metabolic disorders and other complications also. They are using, Indian systems of medicines are used for the all the diseases. Next. So how we have to develop the drug from the raw material to the finished product? So herbal drug development, first we have to select the plant drug. First we have to go for the intensive literature survey. Then we have to find out the plant must be the having according to the Ayurveda system previous literatures. We have to select the plant. Then after that we have to go with the authentication, we by using various botanical taxonomies, then collect the plant material. After that various processing technologies, we have to process the plant material. Then we have to go for the extraction. Here we have to extraction process depending upon the phytochemical, which and depending upon the concentrating on the disease what we have to concentrating. In that case we have to go for the solvents which we have to use it for the extraction of the, and also we have to use the various method depending upon the phytoconstituent which we have to extract. Various extraction procedures are there: cold maceration, and also percolation, soxylate, and also various sophisticated instrumentations available in soxylate, and also microwave fluid extractor, and so many other supercritical fluid extract. So many extractive processes are available depending upon the phytoconsistent which we have to isolate. And next, after extraction we have to go for the purification. We are using various chromatographic techniques, HPTLC, and the classical method is column chromatography, and also nowadays we are using um, various gas chromatographies, LCMS, HPTLC, HPLC, LCMS. These all various chromatographic techniques we are using to separate and purification of the phytoconstituents. Then we have to go for the pure single component by using the column chromatography. We have to go for the biological evaluation, clinical screening, and also various clinical studies we have to go. And finally we have to process the phytomolecule into the formulation. And depending upon the nowadays, and uh, computer aided drug design is also coming to the picture. For before we have to go for the preclinical studies, so we have to go for the cat, then we have to go for the clinical studies, then preclinical and clinical studies, then we have to go for the formulation of the drug. Next. So why herbal standardization is necessary? Till now we are discussing about the what is the herb, how the herbal drug technology revolves, and how we have to develop the herbal formulation. Before we have to do all these things, why we have to go for the herbal standardization? Standardization is very very important because internationally increasing the faith and demand of the herbal medicine, because there is a lack of acceptability of our Indian systems of medicine in the few foreign countries, because due to the lack of standardization protocols, and there is a rigid rigid standardization profiles is not available till now due to lack of technical documentation, lack of approaches in development of modernized dosage forms of herbal medicine, lack of specific process for manufacturing of the formulations, lack of routine quality control methods for finished preparations. So herbal remedies are being used throughout developing and developing countries and representing the sustainable proportions of the global drug market. So it is therefore essential to establish the standardization rigid quality control protocols to accept our Indian systems of medicine in the international and global market. Next. So what are the advantages of the herbal standardization? Because first thing is industry institute linkage, we get the quality assurance, and we have batch to batch consistency of that particular formulation, increase expert potential, desired therapeutic effects we can achieve, acceptability in the international market increased by the many folds. Okay, these are the various benefits of the herbal standardization. And how herbal standardization have the international demand? Because a international pharmacopoeial federation recommends the biopharmaceutical characterization of the herbal medicinal products, and WHO encourages and recommends the traditional herbs remedies in national healthcare program, and various world health organizations also there to ensure the quality of the medicinal plants products by using various modern analytical techniques applying suitable standards. And before we have going to the various methods of the standardization, we have these three are very very important: quality, safety, and efficacy of the every herbal formulation. So these are the various steps of standardization. First step is raw material. Next one is raw material identification and also evaluation. Second thing is process standardization, and third one is product standardization. In the raw material standardization and evaluation, first we have to go for the authentication of the raw material. This is very very important. Nowadays all the herbal industries face a challenge because there is a standard there is a pure raw material is not available because due to the various adulterations. Before we have to go for the collection processing, we have to go for the authentication, and also we have to use various evaluation protocols to assure the plant material is genuine or it is a quality and pure, and then go for the collection and processing of the raw material, then go for the various physicochemical evaluation parameters. And in this physicochemical parameters we have to go for the extractive value, ash value, and also various TLC, HPTLC, phytochemical screening. It will we have to do all these evaluation parameters under physicochemical evaluation. Next, go for the process standardization. In the coming to the raw material standardization, we have to go for the collection of the plant in which season, region, time, drying time, preparation, storage of the these all things influence the quality of the final herbal formulation. Next. And these are the various physicochemical evaluation parameters. Those are macroscopy and sensory evaluation, that is organoleptic evaluation, macroscopy and microscopic, as value, extractive value, volatile matter, bitterness, swelling index, foaming index, radioactive contamination, pesticide residue, heavy metal contamination, microbial content, and also we have to do the various chromatographic profile, which type of compounds are present in that particular raw material by using TLC, HPTLC, moisture content, foreign matter. These are all the evaluation parameters are helpful to get the purity and quality of the raw material. Next, coming to the process standardization, here we have the process uniformity and in-process quality control. Here we have to do the authentication of the plant material shade and mixing, when we have to mix, in which temperature we have to do the processing, time of the processing, and humidity. All these parameters we have to take into the consideration while standardizing the raw material for the process. And coming to the in-process pH, viscosity, depending upon the formulation what we have to prepare, accordingly we have to follow the various protocols from in this in process standardization, and we have to maintain the batch to batch uniformity and consistency. Then coming to the quality assurance of the finished products, these are very very important. After final formulation also we have to go for the various evaluation parameters. These are extractive value, content uniformity, TLC profile, heavy metal content, and also pH, ash value, and moisture content. These are the parameters, and these are the evaluation protocols are necessary to evaluate the purity of the final formulation. These are the various formulations, tablets, pills, depending upon the herbal formulation what we have to prepare, depending upon the Siddha formulation and Unani formulation, or either herbal formulation. Depending upon the formulations we have to change the evaluation parameters. Accordingly we have to do we will get the quality and purity of the final product. And this is very very important, that is fingerprinting evaluation of the fingerprinting and quality evaluation of the herbal formulation. In that we have to see the marker component and also the extract. We have in this fingerprinting analysis we are using various TLC, HPTLC, GCMS, and also HPTLC and LCM, HRLCMS. These all chromatographic techniques we are using, whether the identify the various constituents are present in that particular extract or not. We have to do either by the quality and quantity also we have to compare it with the standard marker component. It is useful when even when the correct component is not there, we have to compare with the standard markers. These are the various chemical markers. Example, oleogum resin, we have to go with the TLC fingerprinting analysis, and we have to do the HPTLC, both qualitative and quantitative also. Next, these are the TLC profiling of the Sirreta. These are the HPTLC. Some chromatograms of the HPTLC. So from the fingerprinting analysis, HPTLC also one of the tool. We have to get the information of quality and the quantity of the particular component which are present in that particular extract, and also from the component formulation. These are the some of the HPTLC data. Next. This is the UV spectroscopy. Some of the these are all the fingerprinting data of the some of the components. Next, coming to the safety and efficacy assessment, this is also very very important of the herbal formulation and pharmacodynamic studies and pharmacological investigations. We have to, even we don't know the actual pharmacological activity of that particular molecule, we will go for the blind screening. Otherwise, depending upon the literature survey nowadays, molecular docking studies, we have to go for the pharmacological investigations. Before we have to go for the preclinical studies, we will go for the toxicological investigation, acute toxicity, long term toxicity, subacute, and also chronic and special toxicity studies. And then we have to go for the in vivo studies, preclinical studies. Depending upon the activity, desired activity what we have to do, then go for the clinical trials, phase 1, phase 2, 3, and 4. And stability assignment is also very very important, not only for the allopathic medicine, that is the herbal drugs are more and more concentrate on stability assignment. Due to this is the easily prone with the microbial parameters. So these are the various stability parameters we have to consider, physical parameters and chemical and microbial parameters. And guidelines and monographic approaches, these are very very important. Till now, why our Indian systems of medicine is not accepted in the foreign country? Due to the lack of rigid quality control tools, and there is no rigid monographic profile for the each herbal drug which are used in Ayurveda, Siddha, or Unani. So that's why we have to as a pharmacognosist we have to develop the monograph profiles for the each component of the herbal drugs, WHO guidelines for the assessment of the herbal medicine, and US FDA and WHO guidelines for the stability testing of the pharmaceutical products, and also WHO guidelines for good manufacturing practices, WHO research guidelines for evaluating the safety and efficacy of the herbal medicines, and guidelines for the quality control methods, guidelines for the various biopharmaceutical characterization of the herbal medicine product, and also European guidelines on the evaluation of the herbal products. These all we have to follow and make a final monograph for the development of the standardization protocols for the each and every, not only for the individual plant material, for all the further formulations. These are the other Indian Pharmacopoeia monograph, Herbal Pharmacopoeia of India, British Herbal Pharmacopoeia, Japanese, European, US Pharmacopoeia, and also various pharmacopoeias that are there to develop the protocols. And these are the various limitations. First thing is chemical complexity, lack of therapeutic. These are the very very important. Because of these limitations only, our Indian systems of medicine is not accepted by the foreign countries. Because first thing is there is no rigid protocols, and there is no evaluation parameters, and there can be the plants which are procured from the one region having the potency of some components are vary from the other region, and also the chemical markers will vary, and so many factors are coming to the picture for limitation standardization protocols. In that case, we have to make a proper monograph for each individual plant material which can be used in that formulation. Intrinsic factor and also extrinsic factors, all these are factors are affect the standardization for protocols. So need for complementary tools for the optimal characterization of the herbal medicine. And coming to the herbal drug translation, not only the raw material and final product, these are the biotechnological approaches also very much useful for the standardization of the herbal drugs. One is molecular markers, DNA molecular markers, and second one is micropropagation. Next. So, what are the various applications of molecular markers and genome analysis and breeding, mapping and labeling of the genes, and generating tools for the marker-assisted selection in the plant breeding, and diversity analysis of exotic germplasm and germplasm preservation by in that way, genotyping of the cultivars, and phylogeny and evaluation. These molecular markers nowadays a very very important tool. We have to identify the various uh, authentic plant material in the molecular level only. So here next generation sequencing is a tool came into the picture to identify the various genes. We have to modify, and we have to develop a protocol to identify the DNA molecules. Accordingly, we have to identify the various adulteration, and also the not only adulteration, we have to identify the in which way we have to control the production of the secondary metabolite. In so many ways we have to control the standardization protocols. These are the various molecular markers, sequence based markers, PCR based markers, hybridization markers. Next. And steps in molecular DNA markers. First, we have to extract the DNA, then PCR amplification, electrophoric separation, visualization, and from that we have to go for the data analysis. So this is the DNA extraction, from the DNA extraction to from the plant, and we have to develop the isolation of the DNA. This is the processor for the isolation of the DNA. From the isolation, after isolation we have to go for the PCR amplification. In this picture we can see, after DNA or RNA isolation we have to go for the biotechnological aspect, reverse transcription, then PCR amplification. We identify the gene along with the codon. We have where exactly. So after data analysis we have to identify the where the plant is exactly showing the same gene or genome. So from that we have to identify the molecular marker, we have to fix that marker. From that we have to develop the protocol. Molecular markers in the standardization, which is used for the authentication and also detection of the adulteration and substitutions, and also it is used for the formulation of various standards like Trifala churna. We are using three types of plant materials. In that way, which type of molecule is it adulterated or not, it is exactly using that particular starting material or not. By using this molecular marker, we have to give the assurance that that formulation must be the pure and standard. Next, and this is a one DNA sequencing characterized by the next, coming to the micropropagation. It is a technique for regenerating clonally uniform plant under aseptic condition. We all know that micropropagation we have to develop into the various explants to develop a new plant, and micropropagation is one of the production of the whole plant for the form small various parts of the plants, from the stem tip node and also the embryo meristem and various little argon seeds, and you have to develop a desired whole plant. So final conclusion, why we have to develop a standardization, why we need the standardization? Because first problem is we have various Indian systems of medicine like Ayurveda, Unani, and also various Siddha systems of medicines. These are various ancient systems of medicine. Even though in not only in the foreign countries, even in our India, no one is not aware about these Ayurveda systems of medicine, because there is a lack of standardization protocols. So for that, and also not only use it for those systems, and also we have to uh, accepted by the globally, we have to take it in these considerations. That is, one thing is we have to constriction of Indian systems of medicine standardization. First we have awareness of Indian systems of medicine. Backslide, excuse me, black back previous slide. Awareness of Indian systems of medicine standards need to be reformed, and also it needs to be improvised. Already some standardization protocols there in WHO, and also Indian system, and also pharmacopoeia, Indian pharmacopoeia, but we have to improvise the systems, because those in that monographs, the components which we have to evaluate is not up to the mark. Nowadays we have various sophisticated instrumentation technologies to the coming to the picture. According to that, we have to develop the protocols. Revised methods of standardization need to be completed. Consolidate the support system of Indian systems of medicine. Vigorously advanced the process of Indian systems of medicine international standards. So India can be emerges as the major country and play the lead role in the production of standardized therapeutically effective herbal formulation. India needs to explore the medicinally important plants. This can be achieved only if the herbal products are evaluated and analyzed using sophisticated modern techniques of standardization. So standardization is very very important when we have to make a good and quality herbal formulation. Thank you one and all. Thank you so much. Thank you so much for giving this opportunity. Thank you, sir, Dr. Achut Kumar, sir, Principal, Srinivasarao College of Pharmacy, for giving this opportunity, and the management of Sri Srinivasarao College of Pharmacy, and the co-convener, Dr. Swati Priya, and also various faculty members who are attending this faculty development program. Thank you so much. If anyone having the queries, welcoming you.
If anyone have doubts, put it in the comment section.
Dr. Arya ma'am, that was a wonderful session. Dr. Arya ma'am, that was a wonderful session. So I have
Thank
a
you, Dr.
small query,
Swatipriya.
ma'am. Like when we are going for a polyherbal extract preparation, like if we are formulating it into a cream, what are the standardization tests or what are the tests which we need to perform?
So you are preparing the herbal formula cream.
Yes, ma'am. Cream.
So like cream, we have to go for the general cream evaluation parameters. Before that, before we have to go with the cream, we have to select the plant material. We have to do the raw material evaluation parameters. So what
Okay.
type of using the powder or extract you are making into the cream. So we have to go with the first raw material evaluation parameters like ash value, extractive value, crude fiber content, all these things. Then we have to go with the extraction process. Then for that extraction, generally cream we have to go with the spreadability, content uniformity, and like that we have to go for the creaming and also cracking test. These we have to go for the general cream evaluation parameters. But before we have to go with the cream preparing the extracts, we have to follow the raw material protocols. But for the evaluation of the content and also the dose, we have to go with the HPLC.
Okay. We need to go for HPLC. Yes. Thank you, ma'am. Thank you so much.
Okay, ma'am, thank you so much. Can
Any
I
more
leave?
queries from anyone? Dr. Arya ma'am, someone raised a question. Explain DNA slide.
Vada ma'am, you can go with that.
Dr. Arya ma'am,
Go. So here
DNA slide.
this is na wait back next back
Next slide.
back back. So no DNA no
Yes.
back whether no back no back. So DNA molecular marker standardization, correct? Go with that slide, Swati ma'am.
సార్ డిఎన్ఏ దగ్గరికి వెళ్ళండి ఆహా, ఇంకా
అక్కడికి నెక్స్ట్ మాలిక్యులర్ మార్కర్
yes, ఒక
ఇంకా
page,
వెనక్కి
ఒక
వెనక్కి
back,
ఇంకొకసారి ఇంకొకటి ఆ సో
ఒక page.
సో
Next.
along we have before standardization protocol, we have raw material standardization is the one standardization, and also finished product standardization. Like that, this is the DNA molecular marker standardization. So here this is very useful to identify the authentication, identify the plant material which we are using for the formulation, it is genuine or not. First thing is authentication, then we have to detect the adulteration of that particular molecule, that particular plant material. So here these are the various steps. DNA extraction, first we have to extract the DNA from the plant, then we have to by using various methods, and then go for the PCR amplification by using various markers, then go for the electrophoric separation, then we have to get the visualization and data analysis. So this method, molecular marker methods are useful for the identification of the particular marker. We have to decide the molecular marker. Next slide. దీని నెక్స్ట్ స్లైడ్ దీని ముందు స్లైడ్ Ah, here in
అదే
next slide.
ఫ్రంట్కి
ఇది
వెళ్ళండి సార్.
కాదు కదా.
నెక్స్ట్.
So DNA extraction, we have to extract the DNA. Then in the DNA, we have to go with the reverse transcriptase. Then accordingly, we have to go with the PCR amplification. From that, we have to analyze the data. So first we have to do the data. If the molecule, the plant material having the gene, correct? Example, Vinca. Vinca is having one gene, so it is containing gene and genome. So we have to extract those DNAs and reverse transcriptase. Then we have to go for the PCR amplification. Then from that data, we have to get if the Vinca rosea gene having this marker. So next time when we have to use that, if that marker is present, this must be there in that formulation. So like that we have to authenticate that particular plant material.
Yes, ma'am. Thank you. We have another query. What are the parameters for standardization of herbal drugs?
We have so many parameters from the raw material to the finished products. First thing is organoleptic parameters, as I discussed. In this organoleptic parameters, color, taste, structure, then microscopic and macroscopic evaluation, microscopic powder microscopy, and also transfer section of the various parts of the plants, ash value, extractive value, and also crude fiber content, and also uh, fingerprinting analysis, uh, total phytochemical, total oil content, phenolic content, and also total heavy metal residue, microbial content. These are all parameters of standardization of herbal drugs.
Ma'am, next query is,
Please
ma'am, please
easy
tell a
way to
easy
convert
way
the
to
liquid
convert
extract
liquid
into
extracts into powder.
go for the freeze dryers, freeze dryer.
So next query. Query, sir. Query. చెప్పేశారు మేడం. Can you please explain the scope of spectroscopic techniques in the standardization of herbal formulations?
Without spectroscopic techniques means NMR, and these are very very important if the extract is having some phytoconsistence. So how we have to know if the extract is having A, B, C? So when we have to go for the column chromatography, we have to isolate the molecules. After isolation, we have to characterized by using spectroscopic techniques. That is for the molecular weight, we have to go for the mass and molecular formula. Determination of molecular weight and molecular formula, we are go for the mass spectroscopy, and IR we are going for the various functional groups, and we have to go for the NMR, proton, and see that in LMR. So these are all spectroscopic techniques are very very useful for the characterization of the phytomolecule which are coming into that particular extract.
Any more queries, please? Yes, thank you. I request Mrs. A. Kanakadurgawalli ma'am to proceed the next session.
Priya, for discussing about ancient traditional pharmacognosy to the advanced pharmacognosy of present era, and giving a clear picture of about chromatographic techniques that are used, and also various stability parameters, and also DNA molecular markers, and how to do the standardization of Ayurvedic products also. And thank you for accepting our invitation, and hope in future we have more collaborations. And
Thank you,
I
ma'am.
thankful to the management for accepting and organizing the FDP. And I thank you, our principal, Dr. K. Achyut Kumar Garu, for being the backbone for organizing this FDP in smooth manner. Thank you. Thank you very much.
Thank you so much, ma'am. Dr. Swati Priya, can I leave, ma'am?
Yeah, leave it. Yeah, you can leave. Thank you very much.